Okay, time for the bio song. Hold Up by the Lady in Lavender, Cassidy Howe, and Lady Minaj, Sadie Burgess. Ready for this fresh beat? Go. Griffith! Try to find the vaccine for pneumonia. Got it straight and put it in the mouse. What happened to the mouse? It died! Second time around with an R strain. Our R strain. Injected, deflected. The mouse is alive. Take that, Griffith. Time to heat kill. Heat kill. The S strain no longer has virulence. What's virulence, you say? That means it can cause disease, even death. You could even say that heat killed S strain now has a virulence. That means that it cannot cause disease. No disease here, and the mice are alive. Yes! But what happens when you put it with the regular R strain? You get some dead mice. This means that the deadly part of the S strain must be incorporated into the R strain. What? Oswald Avery, not the octopus, repeated Griffith's experiment. Copycat. But Oswald put protein-destroying enzymes in one batch, Uno, and DNA-destroying enzymes in another batch. The trial with protein-destroying enzymes still underwent transformation, which means that protein can't be the genetic material. What is transformation? It's when you take DNA from one organism and put it in another. What? The trial with enzymes that destroy DNA didn't transform. This proved DNA was the genetic material. Go, Oswald! Take that, science! Ready? Okay. Done by Alfred Hershey and Martha Chase. They wanted to find out what the genetic material could possibly be. Protein or DNA. Ready? Science! They started with a batch of a virus with radioactive sulfur. Sulfur is in proteins, but not DNA. Important. They made another what? Another batch of virus with radioactive phosphorus. Phosphorus is in DNA, not proteins. They take each batch and let them infect separate cells. Now it's time to blend. Blend each batch separately and measure the radioactivity of the cells. Cells with radioactivity had DNA inside of them. What does that mean? Proves that DNA is the genetic material. Go Hershey and Chase! Take that, science! Now we're gonna learn you some DNA replication. Replication. We start with the DNA helicase. What does that do? Separates the strands that are inside of you. But what keeps strands from reconnecting? Why, that would be the single-strand binding proteins. DNA polymerase is an enzyme that does most of DNA replication. It uses hydrogen bonds, which are bonds between polar molecules, to pair the complementary bases with free nucleotides. But wait, there's more. It also attaches neighboring nucleotides to each other with covalent bonds. That's a bond done by sharing electrons. But would you believe that there's even more? DNA polymerase also proofreads and fixes mistakes. Thanks, polymerase! What? Transcription! That's the process that converts DNA into RNA. Hold up! Those are some big words. Let's define a few. Rapid fire, go! Talking about ribonucleic acid, hereditary molecule from species, ribonucleic acid, working copy of your DNA. First RNA polymerase binds to the promoter. RNA polymerase then splits the DNA strands. Nucleotides of the RNA bond with complementary DNA bases that are exposed. Uracil and RNA pyrimidine replaces mean. Still rapping about RNA polymerase. Ace. So listen up, yo. Now it attaches neighboring nucleotides into just a single strand. Whoa. Hold up, Lady Minaj. RNA polymerase will stop transcription around her termination sequence. Now what? The messenger RNA, aka mRNA, leaves and the strands go back together. Translation. What's that? It's the process that turns RNA into protein. No way. First out mRNA binds to ribosomal RNA, which we also called rRNA, to attach to the ribosome. But wait, what's mRNA? What's rRNA? Rapid fire, go! mRNA brings the DNA part of the ribosome. rRNA makes it part of the ribosome. Time to get some transfer RNA, aka tRNA. But hold up, only with the appropriate anticodons and amino acids. Now let's take that tRNA and bond it to the codon of mRNA in the P site. PP site. Hold up. Now you repeat with the second tRNA, but in the A site. AA site. Now a peptide bond. What? A peptide bond, a bond between amino acids, forms between the two amino acids. Now a ribosome translocates. Yeah, ribosome translocates. First tRNA is now in the E site, EE site. Second tRNA is now in the P site, PP site. First tRNA leaves the ribosome, kicked out. Process repeats until the stop codon, hold up, reaches the A site. AA site. Protein leaves the ribosome. All done. Fire mixtape, hold up. Bio. Learning.